Okay, so we're going to look at transport in plants, right? As a chapter, new chapter here. So what are the things you're going to cover according to the syllabus is these are the learning outcomes. So the one we're going to cover today are the one that is in darker color. The gray color one, we will do it during practical session when we have opportunity to go to the lab. Okay, right. So today we're covering number 2, describe the distribution of xylem and phloem in the herbaceous dicot roots, stems, and leaf. Okay, so that means you need to know where is the location of xylem and phloem when you're given herbaceous sample of dicot. That means there are dicot and monocot, right? So you only need to know dicot. You don't have to know monocot. And then herbaceous plant means non-woody plant. Okay, so given the roots, given the stem, given the leaf, transverse section, either in the form of a slide or in the form of a micrograph or in the form of a diagram in MCQ question, structured question, practical exam, you need to be able to identify the location of xylem and phloem. Okay, then relate the structure to the function of xylem vessel elements, sieve tube elements, and companion cells. So these are the 3 important cells that play the role of transportation in the plant. So this is what we're going to cover today. Okay. Transport system of the plant is called the vascular system. So vascular system comes from the word vas. So vas actually stands for vessel. So it is a system made up of a lot of vessels or a lot of tubes. Okay, tubes. So when you want to transport material, you transport it with tubes, right? Like transport water with water pipe, right? Water pipe. And transport our blood in a blood vessels, right? So transport vessel, vascular system. Now the vascular system is divided into 2 types of tissue. It's made up of 2 types of tissue. It's either xylem or phloem. So when you say xylem and phloem, you are referring to the tissue. Now remember that when you learn tissue, right? Well, what is the definition of tissue? Philbert? What is the definition of tissue?
A group of similar cells that perform a specific function.
Characteristically arranged and perform specific function of the tissue. So tissue is a group of cells which may not be similar. It may be or it may not be similar. Okay. Characteristically arranged means they will arrange in a certain kind of arrangement, and all of them will perform the specific function of the tissue. So that's definition of tissue. So a group of cells. So far, we learned that tissue is made up of a group of cells that are similar, right? Like for example, adipose tissue is made of fat cells, right? Our skin is made of epithelial cells, right? So most of the time when we learn the tissue, we learn that the cells of the tissue are the same type. But over here, you see xylem and phloem tissue, each of them is made up of 4 different types of cells. Very interesting, 4 different types of cells. So therefore, tissue's definition is a group of cells, may not be similar. And these 4 different types of cells, they are characteristically arranged, and all of them group up together to carry out the specific function of the tissue. Now, each of the cell will play a different role, but all of them group up together in order to achieve the specific function of that tissue. Okay? So, among the 4 cells, xylem is made up of xylem vessel. Xylem vessel element, okay? Sclerenchyma, parenchyma, and trachate. Phloem is made up of sieve tube element, companion cells, parenchyma, and sclerate. Sclerate can also be called the stone cells, or sclerenchyma. So you can see now that a tissue sometimes may be made up of more than one type of cells, like xylem and phloem tissue. Okay, and also please learn up the name. Whenever you say xylem and phloem, you are referring to the tissue. So what exactly is the cell that will transport the water is actually the xylem vessel element. What exactly is the cell in the phloem that will transport the sucrose is the sieve tube element. Okay, so please learn up the name. So the xylem vessel element, they will join end to end in order to form the xylem vessel. So when you say xylem vessel, right, the long tube that transport water. Now, this long tube is made up of individual cells that are joined up end to end. So this is one cell here. So I'm just to show you. So this is one cell here, right? So the end of this cell, so this is the end of the cell, right, will join up to the end of another cell. Okay, so this is the xylem vessel cell. Now on top and bottom is empty one, because it's supposed to join up to form a long tube, right? So this is the end of this cell here. So this cell got two ends, the top and the bottom end. And this cell also got two ends, the top and the bottom end, right? So the end of the cell will join up end to end to form a very long tube. So this is a so-called xylem vessel. So when you say xylem vessel, right, you are referring to this tube. And this tube is made up of many xylem vessel elements that are joined up end to end in order to form the xylem vessel. So therefore, you need to pay attention to xylem vessel element is the cell. Xylem vessel is many, many cells joined up to form the vessel. Okay, so same as sieve tube elements will join up end to form the sieve tube. Okay, so now we want to look at the function of the xylem. That means you're referring to tissue. So what is the function of the xylem? Xylem is for the transportation of water and dissolved mineral ions. So you know that the water comes from the soil, and inside the soil got a lot of mineral ions, right? Like nitrate, ammonia, iron, phosphate, etc. So all these mineral ions will dissolve in the water and therefore can be transported together with the water into the plant body. Okay, so xylem will carry them. And also xylem's function is for mechanical support. Okay, mechanical support means physical support. Later on, you know why. Okay, so now we need to relate the structure of the xylem vessel element. So when I say xylem element, I'm referring to the cell. Element means what? Elementary means basic. So the basic structure of the xylem vessel is the cell. So basic, isn't it? Which is actually the cell. Okay. So xylem vessel element or xylem element is actually the cell. So we're going to look at the structure of the cell and relate the structure to the function. Okay. So number one, it is hollow. It has hollow lumen and there's no cytoplasm inside the cell. So when the cell is supposed to transport material, you want the cell to be empty in the center. So there's no cytoplasm at all, okay? So that it will reduce resistance to the flow of material. So the cell is totally empty. It's only made up of cell wall and nothing else. All the living part of the cell are not there. So that's why it is a dead cell. So because it's a dead cell, there is no cytoplasm. Okay, no cytoplasm, no nucleus, no nothing. It's a dead cell. Only left the cell wall. Okay, so why? To reduce resistance to flow of material. So structure function. And then number two, there are no end walls. So in a normal plant cell, when the cell is newly formed, there must be cell wall, right? So let's say, for example, now this cell is newly formed. That means after mitosis and cytokinesis. So you got a cell, and the cell is usually square in shape. So after cell division, in order to form the xylem vessel element, the cell will have to go through cell elongation, followed by cell differentiation, right? So this is a process of how the cell will grow, right? So after cell division, the cell that's newly formed is square. Then it will elongate and differentiate in order to form the specialized cell, right? So during specialization, okay, during elongation and differentiation, then the cell will start to take up specialized structure and function. So as it elongates, it will also synthesize things that are specialized for the cell. So that's why during elongation and differentiation, specialized structures will be formed. So in this case here, what happened is that the end walls will be removed during specialization. Hang on. So the end walls will be removed during specialization. So this wall will be gone, right? And then additional wall will be formed, which is the secondary wall. So additional wall will be formed here. Can you see? You cannot see. Okay. Let me use solid color. So additional wall will be formed here. Okay. Additional wall will be formed here. Can you see or not? The red color wall. So the red color wall is the so-called secondary wall. So the green color wall is called the primary wall. Remember, you have learned the cell wall is made of primary wall and secondary wall. So the newly divided cell, the cell wall is a primary wall. When the cell specializes, it will form the secondary wall. So this secondary wall is made up of cellulose, hemicellulose, and one additional thing here, which is the lignin. So this is the main thing. It's lignified. Okay, the secondary wall is lignified. So lignin is rigid, so it provides a mechanical support, a better support to the wall. Okay, and is also impermeable to water. So lignin has a property that is rigid, so it provides mechanical support, and it is also impermeable to water, so the water cannot move in the cell wall. The water must move inside the lumen of the cell. It will not be moving in the cell wall because lignin is waterproof. Okay? Right, so now I'm showing that now end walls are not there. So the cell has no end walls. Why the cell has no end walls is because then the cell can join end to end. So this is one cell here. And then the end to end can be joined up to form a very long tube. A long continuous tube. So that's why the tube can be continuous, because there are no end walls. Okay, so no end walls so that the cell can join end to end to form a long continuous tube to transport the water from the root to the stem to the leaf continuously without obstruction. Okay, so now why the wall must be lignified? I already said, to provide mechanical support, right? So why do they need to have got mechanical support? It's because the tube is used to transport water. Now, how is water transported up the plant? What mechanism causes the water to move up the plant? Xin Yin Ding.
Yes, teacher.
Yes, resume recording. So water is transported up the xylem vessel by transpirational pull. So transpirational pull is a suction force. So imagine the xylem vessel is like a straw, and then you're trying to suck up the water from the straw. Water using the straw. You're trying to suck up the water in your glass using the straw. So you know that nowadays are all sorts of straw, right? So imagine you've got a straw that's transparent, the very simple type, that is quite thin and flexible, the wall. So if you use a force to try to suck up very strongly, instead of sucking up the water cannot move up because the wall of the straw collapses, right? When you suck, it's so strong that you pull the both walls together and they touch each other, and then the water cannot move up anymore. So there is this danger in the plant. Because in the plant, the xylem is a continuous from the root to the stem to the leaf. Let's say the plant is very tall. So the xylem vessel will be very, very long one, you know, all the way from up to down, all the way to the root one. And then you've got a transpirational pull up there, and daytime when the sun is there, evaporation is very strong, transpirational pull becomes very strong. So if the wall is very thin and not strong, when the transpirational pool is very strong, the wall is definitely going to collapse inwards, right? Because of the suction force. So the wall is going to collapse inwards if the wall is not strong and it's very thin. So we say that the wall may collapse in the daytime due to transpirational pool. So then, even though there's a pooling force, water cannot be sucked up from the soil up to the plant already. So it defeats the purpose of transpirational pool. So therefore, the lignin is added as a secondary wall to the cell to strengthen the wall so that it provides mechanical support so that the wall will not collapse due to transpirational pool. So that's why the wall of this xylem vessel is lignified. It is to prevent the wall from collapsing due to transpirational pool. Okay? So the cell is not only have got empty lumen and hollow lumen and no end walls, the cell will join end to end to form a very long tube. So allow continuous flow of water from the root to the stem to the leaf without obstruction. Okay. Now, one more thing that's very interesting here is, do you realize that I draw the wall with pits, right? So these are the pits. Okay, so you've got a lumen, you've got a lignified wall, and you've got the pits. So you need to do the labeling in your notes. The pits are very, very important because you know that when you transport the water from the root to the stem to the leaf, right? So let's say, for example, it's traveling up the stem. So this is the plant. And then you've got the roots, right? The roots. Okay. Then the xylem will start from the root, isn't it? Okay, the xylem vessel. Then it will go up the stem this way. Right? After that, you'll go to the leaf wherever. Now, you know that it's traveling up the stem. The stem is not only made out of xylem vessel. You learned before that the stem has got a lot of other cells. So let's say, for example, I draw the stem, okay? So you know that the stem looks like it's a transverse section, right? It will look like this, right? With a lot of cake-like structure in the center here. So the xylem is located here, right, in this region here. So this is the xylem tissue. I'm talking about the tissue xylem, okay? So the xylem is located here. So around the xylem at this area, there are a lot of other cells. You name it, you've got the epithelial cells, you've got the cells in the cortex, you've got the cells in the phloem, the cambium, and then in the pith. All are made out of cells. So those cells will also need water. Every cell in the plant will need water, right? Because you already learned the function of the water. Can you tell me what is the function of the water, Jing Er?
Yes, teacher.
What is the function of water for the cell? Recording. Okay, so the function of the xylem vessel is for lateral transport. So as the water is transporting up, you have to move to the sideways to supply the surrounding cells with water. Okay, so this is lateral transport. Then second function of the pit is to bypass airlock. Airlock means sometimes there could be a column of air that's being trapped within the xylem vessel. Why? Because sometimes the soil is totally dry, but there's transpirational pool. So therefore, a column of air will be sucked into the xylem vessel, and then after that it rains, and then the water is in the soil and the water is being sucked up. So you can see that here, this is the a column of air inside the xylem vessel. So therefore, when it's column of air like this, right, the water cannot move up anymore. Okay, this column of water here cannot move up the xylem vessel anymore, because this column of water is not connected to this column of water. So it will not be pulled by the water column here upwards, because water can move up because there is a extensive hydrogen bonds among the water molecules to pull them up, right, all the way until the leaf. So wherever the water move and evaporate, the rest of the water in the xylem vessel will be traveling up together. But over here, this column is not connected to this column by hydrogen bond at all. So that's because of that, the column of water here cannot move up anymore due to the airlock. Okay. So therefore, it has to, in order for this column of water to move up, it has to go out from the xylem vessel through the pits like this one here, and then move up the surrounding cell, and then move in here again through the pits. So move up and then move in again through the pits. So because surrounding the xylem vessels, there are a lot of other different types of cells that are not hollow. So there are cytoplasm in it, so definitely got water in it. So this water in the xylem vessel will move up to the neighboring cells and then go back into the xylem vessel. So that's how they can bypass the airlock. Okay. So label the structure lumen, lignified wall, and pits. So let us look at the lignified wall. Okay, a little bit detail of the lignified wall. It's very interesting. You see the lignin thickening, right? Lignin thickening is a secondary wall, right? So when you add the secondary wall, the cell wall becomes thicker, right? So that's called the secondary thickening. So they add lignin, so it's called lignin thickening. So you see the lignin is very interesting. The lignin can be annular. So that's how they put the secondary wall. So annular means ring form. It can be spiral. It can be scalary form. Scalary form means it looks like a ladder. So scalary means ladder-like. Okay. Okay. Reticulate means net-like. Reticulate stands for net. So you see, reticulate is net-like. Pitted means the entire wall, a secondary wall, is made of lignin except for the area with the pits. The area with the pits don't have secondary wall, but is there a primary wall there, Samir?
Yes.
So there's a primary wall, right? Because just now I showed you that there must be a primary wall as the first layer of the wall. Then the secondary wall, which is the lignin, will be placed between the primary wall and the cell surface membrane, right? So the secondary wall is placed here. So wherever it's pitted, that means there's no lignin, right? Wherever there's pitted, there's no lignin. So if you say there's no lignin here, there's still the cellulose wall, the primary wall is still there. And the primary wall, the cellulose wall, is freely permeable. So when you refer to the pit, it's actually this area here, this whole area. No lignified wall, but got primary wall present. Okay, Samir got 1 mark, not 1 mark, 2 points. So give Samir 2 points. Okay. Okay. So the primary cell wall is made up of cellulose, hemicellulose, and pectin. Secondary cell wall is made up of cellulose, hemicellulose, and lignin. Okay. So now we look at the secondary thickening, the lignin. It can be annular, which is ring form. Spiral means it's like a spring. Scalary form is like a ladder. Reticulate means net-like. And finally, pitted. Okay. So on the right-hand side is the micrograph of the, I think this is a stem. Micrograph of the stem, which is longitudinal section. So you can see this cell here, right? The diameter of the cell is quite big compared to the neighboring cells. Okay, and you've got the annular thickening. This one you've got pitted. Can you see that? This one has got annular thickening. Now, can you see this cell here? Now, all the pink color is pink color because the lignin is being stained by a stain that will give the lignin pink color. So if you don't stain it, you will not see pink color. You see pink color because it's being stained. So you cannot be going around the plants in your garden and you go and take out the plant and expect to see pink color for lignin. It's not there. You have to stain it, then it becomes pink color. Okay, so then you know that, or the wall has got lignin, so it's pink color. Then you easily identify the cell. Okay, so let's move on. Can you see this cell that's pink color? And the diameter of the cell is very narrow one compared to this one. So this cell is not the xylem element. Okay, this is another type of cell in the plant which also got lignified wall, and this cell is called the sclerenchyma. Sclerenchyma. So please learn this up. So other than xylem vessel element, another type of cell in the plant that may contain lignified wall is the sclerenchyma. Sclerenchyma is also called a fiber. It's because longitudinal section, the cell is elongated with tapering ends. Right? Tapering ends means the ends are sharp. Right? Tapering ends. So therefore, it looks like fiber, right? Fiber is something like a thread that's very thin and long. So this cell is thin and long with tapering end. So it's called the fiber. Actually, it's called sclerenchyma cell. Can you see the wall has got lignin with this pink color? And then you can see the tapering end at some region. Not really very clear. I think here you can see this part here is tapering end. Can you see this sharp end here? Right, tapering ends. Right, so this is the sclerenchyma cell. Right, okay. So this diagram you can see transverse section and longitudinal section, TS and LS. LS, this is my LS. LS. Okay. Okay. Right. So you can see annular thickening, reticulate, peated, okay, scalariform. So how does it look like in transverse section? You can see obviously the wall is thicker than the neighboring wall. Hello? Okay, you can see that the wall of the xylem vessel element is thicker than the neighboring cells, the wall of the neighboring cells. So remember, xylem is a tissue made of 4 types of cells. So in this tissue, you will see different types of cells. So that's why you can see the xylem vessel element plus the neighboring cells which look different from it. Right, so the diameter of xylem vessel is very big compared to a neighboring cell, and the wall is thicker compared to neighboring cells. Okay. So can you identify the xylem vessel now in the xylem tissue? Easy, right? Okay. So then again here, you see here, annular thickening for this one. Okay, annular thickening, annular thickening. This one looks like it is it could be scalariform, which is a ladder-like. Now, why is it empty in the center? Because we happen to section it through the lumen of the cell. So the lumen of the cell is empty. That's why you see empty space here, right? Totally empty space because you're looking into the space. Okay. Now, can you see the neighboring cells that are very small? So these are the other type of cell that you can find in the xylem tissue. Okay. So these are the one that I say that I will convert it into type version. Right? So this is the notes that you want to read because you need to be able to relate the structure to function. And I have also added, other than the xylem vessel element, I've added the other 3 types of cells you can find in the xylem. So I've added the structure and function for your own reading pleasure if you want to know. Right? Sclerenchyma is the fiber. What is parenchyma and what is trachea? Okay, parenchyma is the most basic cell you can find inside the plant. It is used as packing purpose. Packing means whatever spaces are empty, you pack it with this cell. So it's parenchyma cell. Okay, so parenchyma cell have got thin cell wall. It's a normal cell only, used for packing purpose. So no specialized structure there. Okay, now phloem. Phloem tissue's function is to translocate soluble organic and inorganic material. Now the word soluble here is very important because when you want to transport the material must be soluble in the water, right? Then when the water moves, the material will be transported together with it. Now, organic material can also be soluble, isn't it? Right? Inorganic material like mineral ions are soluble because they're ions. Now, can you give me an example of organic materials that are soluble? Can organic materials be soluble? Can you give me an example? Huimin?
Hydrogen.
Hydrogen is organic? No, hydrogen is not organic. Organic means at least it got carbon, hydrogen is organic, right? Not hydrogen. Hydrogen is an element. Okay, should I open to the floor? Yes, open to the floor. Give me organic molecule that is soluble, so it can be transported. Lipid where got soluble? Hey, lipid soluble in water ah. Because you go to your kitchen and put 1 drop of oil into the cup of water and see it can dissolve or not. Cannot, right? Glucose, yes. Amino acid, yes. Alcohol, yes. All are correct. Glucose is soluble, yes. Lao Huiyan. In plant, when the phloem translocate materials, so the materials must be soluble. So both soluble organic and inorganic materials will be translocated in the phloem. Okay, so the organic materials are the assimilates, right? Assimilates. So example, sucrose. 90% of the translocated materials are sucrose. This is the most important thing, but there are also amino acids and vitamins. So all organic materials translocated are produced by the plant. They are all called assimilates. Okay. So what are assimilates? Assimilates are the organic molecules that are synthesized by the plant in a simple structure, so that it's small, so that it's soluble, so that it can be transported. Okay. So then after transported, it goes into the cell. You can use these molecules and convert it into a more complex molecule. Right? So assimilates are molecules that are synthesized by the plant that are simple molecules. Therefore, it can be soluble and can be transported. Right? So there are also inorganic molecules transported because the mineral ions, right, in the soil that dissolve in water transported into the xylem vessel. From xylem vessel, it can cross the wall and go into the sieve tube because cellulose wall is freely permeable. Right? So therefore, from xylem vessel, those mineral ions can cross the wall and go into the sieve tube. So therefore, the sieve tube will also contain mineral ions. So translocation occurs in the sieve tubes. So in the sieve tube, the material can be transported either up or down the sieve tube. So therefore, in the phloem, there are many sieve tubes. All right, so I'm going to draw, let's say this is one sieve tube here. Right, this is another sieve tube here. Okay. Now, the sieve tube may transport material either up the plant, so it can be up the plant, or it can be down the plant. All right. So therefore, this is like you go to the highway. You got one side of the road, the traffic will move up, and the other side, the road, the traffic will be moving downwards. So sieve tube also function in the same manner. Every sieve tube only got the movement in one direction, but all in all, there'll be movement up as well as movement down the plant. Okay. The reason is because if this is a leaf that produces the sucrose, after photosynthesis, convert the glucose to sucrose to be transported in the plant. And don't forget, the plant, right, above this leaf, you've got cells that will need the sucrose, right, because they're still growing. And then you go down the plant, also there'll be cells that are alive that will need sucrose. So here, this is the tip of this stem. The cells are still dividing over here and elongating and differentiating, right? And then you may have got a flower over here. They will also need nutrients because after fertilization, it will form the fruits. So the fruits, you need to supply the fruits with the sucrose. You need to supply the fruits to the meristematic area for the cell to grow. So above the leaf, you will need sucrose. Below the leaf also need the sucrose because the cells here are all alive. So it needs the sucrose for respiration to produce ATP and also maybe to produce other substances in the cell. So therefore, sucrose have been transported up and down the plant, not only down the plant. Okay. So the transport of the materials in the phloem sieve tube requires ATP that is produced by companion cell. So that's why companion cell is always found beside the sieve tube element, because they supply ATP for the translocation process in the sieve tube element. Okay, what is this one here? So sieve tube element or sieve element, they are cells that are alive and they are elongated, and the nucleus has disintegrate during development, just like a red blood cell. Main function is transportation. You want the entire space of the cell to be used for transportation. You remove the nucleus so that it will not block the space. So same as for the sieve tube. Okay. So this is a structure of sieve tube. I would want you to draw this one and label fully. So please draw this one and label fully. And you need to pass up on Monday next week. Okay. So this is your homework. So let us look at the structure. So I'll explain structure and function at the same time. Okay. Okay, structure. So this whole stretch here is these are the sieve tube elements joined end to end. So this is one sieve tube element. And then this one here is the companion cell. Can you see the companion cell, which is always associated with the sieve tube element side by side? Okay, good friend. Now, middle lamella is the middle plate between two neighboring cell wall. So it's in the center of two cell wall. Okay, cell wall, cell surface membrane, sieve plate. Now, sieve plate is the end walls of each sieve tube element. So the end walls of each sieve tube element is called the sieve plate. Okay, why is it called sieve plate? Because this wall is perforated with pores. Perforated means got holes. So therefore, it looks like the sieve, right? It looks like a sieve. You know, sieve is like a net. Sieve is like a net where, let's say you want to make coffee or you want to make tea. You don't want the tea leaves or the coffee beans to mix with your water when you want to drink it. So you will sieve it with the sieve first to filter away the leaf or the coffee bean. Then you just drink the liquid. So this is called a sieve, right? So the sieve plate means the end wall here is perforated, so it's called the sieve plate. Right? Why is it called plate? Because it's flat. Now, in the center of the sieve tube element is empty, so that it reduces resistance to the flow of materials when it's being translocated. Please ignore the word sieve P protein. You don't have to know this one. Okay? Now, there's a nucleus in the companion cell and with dense cytoplasm. Dense means highly compact because the cell is very, very small. So therefore, it will be highly packed with a lot of organelles in it. There are many mitochondria because it's supposed to produce ATP and many other organelles. And then there is plasmodesma. Now I'm going to cancel the ta away because it's one plasmodesma. So there'll be plasmodesma that will connect from companion cell to the sieve tube element. And then the cytoplasm of the sieve tube element are all pressed to the side. So it's called peripheral. Peripheral stands for at the side. Okay, peripheral means at the side. So the cytoplasm are all pressed to the side so that the center is empty, so that it will reduce resistance for the flow of material. And apart from that, there is no nucleus, no vacuole, no ribosome. Because after the cell is mature, you don't want the nucleus to be there to block movement. You don't want vacuole to be there to block movement. You don't need ribosome anymore because the cell no need to produce protein anymore. The cell just have to be there for the translocation of material. So ribosome is not there anymore. Okay? Callose. Now, what is callose? Callose is this layer of substance. Can you see this layer of substance that surrounds the sieve plate? So this is called the callose. Now, the callose end with the word ose. So because it ends with the word ose, it is a carbohydrate. It's like sugar, like glucose, fructose, maltose is a sugar. Okay. So why is there a callose here? Callose function is like blood clotting. It's like a blood clotting function, right? So what happens like this? If there's an injury somewhere here, right? So there's a cut, okay? There's an injury here. So what happens is the transported material, the precious sucrose, amino acid, vitamins, everything might start to leak out from the sieve tube. So just like you cut yourself and then the blood starts to flow out, right? And then all the oxygen, nutrients transported by the blood will be lost from your body. So we have to prevent the leakage of materials that are being transported. So therefore, when there's a injury that caused the leakage of materials, then we have to stop the leakage. So the plant stops the leakage by producing callose. So the callose will grow bigger and bigger, and eventually it will block the entire sieve pore. So this is called the sieve pore, right? The pore on the sieve plate is called the sieve pore. You block the whole sieve pore, then the materials cannot flow anymore. Actually, it blocks the sieve pore here. So the sieve pore here will be blocked, and therefore there will be no more flow of materials. So therefore materials will not leak out from the point of injury. So that's the function of the callose. Okay, so very wonderful. Then, of course, there will be cell surface membranes surrounding the cell wall of the sieve plate. There will be cell wall at the sieve plate, middle lamella, and also the sieve pore, right? So sieve pore, if you look from the top, it looks like this, okay, with a lot of pores in it. The sieve pore is in it. So this is a sieve plate. This whole thing is a sieve plate with a lot of sieve pore in it. Okay. Now my question to you is, you know that when you got a sieve plate like this here, it will actually increase resistance to flow of material, because every time when the material flows here, it will have to, you know, this is like traffic jam. When you got a bottleneck, right? You got a 4 or 5 lane highway, suddenly become 3 lane. Definitely got traffic jam, right? So this will actually increase the resistance to flow. Correct or not? So if you increase resistance to flow, how come it's still there? That means it plays a very important role, or else it will not be there. So what is the role that it plays in the sieve tube? It's like this. Material transported in the sieve tube, right? This is sieve tube, right? Which extend from the leaf to the stem to the root, okay? It's very long. So the material that's transported in the sieve plate is transported by high hydrostatic pressure. So it is the high hydrostatic pressure that push the material to be transported inside the sieve tube. So this is opposite from the xylem vessel. Xylem vessel water is transported by transversational pull. So that you take the syringe and you pull the plunger, you create the suction force. Now in the sieve tube, you take the syringe and you push the plunger. So you're creating a pushing force. So it's the same thing, but it's opposite direction. So if the wall is not strong, right? If you push very strongly, you create the pushing force, right? The wall will actually stretch outwards if the wall is not strong. Can you see that? It will stretch outwards like this. So when it stretches outward, it's going to push against the rest of the cells, the neighboring cells. So we say that the wall may collapse outwards because of the high hydrostatic pressure. Okay, so how to prevent this from happening is if we can hold the two walls together at a certain junction, like this, right? Using the sieve plate. Then at a fixed distance away, we have a sieve plate. Then we will prevent the wall from collapsing outwards because of the high hydrostatic pressure. So therefore, the sieve plate is there to hold the wall to prevent the wall from collapsing outwards due to the high hydrostatic pressure. Okay. So let us look at the micrograph of the sieve tube. Very easy to identify because it looks like our bone. You know the bone? The bone. Okay. The bone. Okay. It looks like the bone like this, with the joints here. So the part with the sieve plate is slightly enlarged one. Can you see here? Right. So the part of the sieve plate is slightly enlarged. I think it's because of the high pressure right over there. So it's enlarged. And then this is longitudinal section. Can you see the cross section here with a sieve plate and a sieve pore? Now the chances of seeing the sieve plate in cross section is very, very slim, because you see when you produce sections to view under the microscope, can you see the entire length of the cell? Maybe only 2 sections out of maybe 20 sections of the cell, you can see the sieve plate. So you try to section the entire length of the cell, right? Only maybe 1 out of 20 of the sections will contain the sieve plate. So the chances of seeing the sieve plate, maybe 20 cells in your slide, maybe only 1 cell got sieve plate. Okay. So this is a sieve tube and this is a sieve plate. Now this is a phloem, right? Phloem is made up of 4 types of cells. So how do you identify phloem? Is very easy. Hexagonal cell that's quite large will be the sieve tube element. Then the neighboring cell that's almost very tiny and triangular, that is called the companion cell. So you can see a lot of hexagonal cell with a triangular cell at the side. Hexagonal cell and a triangular cell at the side. Okay, hexagonal cell with, this is not triangular, but it's angular. Angular. Angular means what? The cell has got certain angle. Angular. So what cells are angular? Triangular is angular. Okay. Square is also angular. Pentagonal is also angular. Okay. Rectangular is angular. Hexagonal is also angular because it's got sharp edges. All these sharp edges is called angular. Okay. So if you don't know what shape is it, but it's angular, you just call it angular. Right? So see here, got a sieve plate with sieve pore, very clear. This is a companion cell because you can see the size of cell so small, right? And it's angular. It may not be always triangular, okay, but it's angular. So companion cell with the nucleus can be visible, right? Companion cell with nucleus visible, right? Companion cell with nucleus visible here. So this is a sieve tube element, totally empty, you can see, but peripheral cytoplasm can be seen, peripheral cytoplasm, okay. Longitudinal section, so this one, can you see this is the sieve tube with the sieve plate, and the neighboring cells are the companion cells, right? Nucleus and then cytoplasm. So this is a vascular bundle with both the xylem and phloem. So how to identify the xylem and phloem? Very quickly, cell, I help you to identify phloem first. So this is a phloem. Why is this phloem? Because phloem is made up of big cells and small cells side by side. So the big cells are usually hexagonal and the small cells are triangular or angular. In this case here, it's angular. So this is my phloem. Then where's my xylem? The rest of them are xylem. Okay, the rest of them here are the xylem. Okay, so xylem got 4 types of cell. Which cell is xylem vessel element? The cell where the diameter is very big and the wall is very thick. And then because it's lignified, the lignin is stained pink color or red color. So can you see this wall is quite thick here? Right, very thick. So these are the 2 xylem vessel element. This is not xylem vessel element, even though the diameter looks very big because it has no lignified wall. Can you see there's no pink color thick wall here? So this is actually an air space. This is an air space because this is not a cell. Okay. Now they also, okay, finish. What else? Now I want you to see here. Can you see this cell here? Wall is also very thick and pink color, right? In this case, here, pink or red color. But the diameter of the cell is very small. So this cannot be xylem vessel element. If it's not xylem vessel element, then what cell should this be? Hoshini. So the next part is you ask to be able to know the location where you can find xylem and phloem in the root, in the stem, in the leaf. So I will show you the diagram, right, in the notes there. So you please read it up yourself. I trust that you have learned this section in your secondary school. Anyone has not learned this one in your secondary school? Please let me know now. I assume that you already learned all this, so I will not touch on it anymore. So you take it as a revision, okay? All right, so you revise again. The cross-section of the stem, how does it look like? Where is the location of the vascular bundle? And where is the xylem, phloem, cambium, and pericycle? Okay, the root. So you need to learn up all the name of the tissues of the root and where is the location of the vascular strand. Now in the root, we don't call this vascular bundle, okay? We call it the stele, right in the center. So in the middle is where xylem is located. This one are the phloem. So you need to know all the layers, okay? So please study as your revision. The leaf, where can you find the vascular bundle? So these are all the vascular bundle here, all right? And you need to know that the xylem is always on top and the phloem is always at the bottom. Okay, xylem is on top, phloem is at the bottom. So how to remember? I think I will remember it this way. The most important photosynthetic cell in the leaf is the palisade mesophyll cell. So palisade mesophyll cell is always at the upper layer of the leaf, below the upper epidermis. So the water must be easily accessible. So xylem on top, phloem at the bottom. This one you have to remember. Okay. So with that, you already learned the distribution of xylem and phloem in the root and stem and leaf. So that's all for today.